tighter wells. There is inverse relation. So it is the general procedure of competitive ELISA, not going in detail. Another type include multiple and portable ELISA. A newer technique uses a solid phase made up of an immunosorbent polystyrene rod with 8 to 12 protruding ogives. The entire device is immersed in a test tube containing the collected sample, and the following step: washing, incubation in conjugate, and incubation in chromogenesis was carried out by dipping the ogives in microbells of initially microplates pre-filled with reagents. Modified ELISA. ELISA. Enzymes will interfere with antigen-antibody interaction. Second antibody is often labeled with a very small molecular substance, biotin, and a specific binding protein for biotin. Avidin is conjugated with enzymes such as HRP. Capture antibody is added, and then target protein will bind to it. Detection antibody, streptavidin or TMB substrate will bind to it. Now coming toward readings of ELISA, how we will read it? Measure the absorbance at 450 with the ELISA plate reader. Calculate the absorbance for each sample and reference. Ascent software for the calculation of results can be used. These are the results. Results may be negative, positive, or assay control. Applications. Since ELISA can detect both antigen or antibody, it's a useful tool for determining serum antibody concentration. It has also been found application in the food industry in detecting potential food allergens such as milk, peanut, walnuts, almonds, and eggs. The other uses of ELISA include a detection of mycobacterium antibodies in tuberculosis, detection of hepatitis B markers in serum, detection of enterotoxin of E. coli in feces, detection of HIV antibodies in blood sample. Now there are certain advantages of ELISA. Reagents are relatively cheap and have long shelf life. It is highly specific and sensitive. No radiation hazards occur during labeling or disposal of waste. Easy to perform and quick procedure. Equipment is widely available. It can be used to a variety of infection. It can be used on most type of biological samples like plasma, serum, urine, cell extract, etc. There are certain disadvantages of ELISA. These include measurement of enzyme activity can be more complex than the measurement of activity of some type of radioisotopes. Enzyme activity may be affected by plasma constituents. Kits are not cheap. Very specific to particular antigen but won't recognize other antigen. False positive and negative possible, especially with mutated altered antigens. Limitations. Results may not be absolute. It's one of the limitations of ELISA. Antibody must be available. If antibody will not available, then this process will not be followed. Concentration may be unclear. False positive possible, false negative possibles. So there are certain precautions that must be followed during this process. These include use of exchange type pipette. Don't use only one type of pipette for whole process. Washing. Wash it very well. Reagents. Reagents concentration should be properly labeled. Reagents. Don't use the same reservoir for multiple reagents. Plate cover. During incubation, well plate should be covered using the plate cover. Plate cover is effective only under suitable condition, that is room temperature. Humidity should be greater than 50%. Airstream should be less than 0.2 meter per second. And last precaution is coating of well. Coating of well should be proper with the addition of blocking solution. Improper coating gave us false positive result. So this was my presentation about ELISA plate reader. If you have any question, so please ask in comment box. Thank you very much.